This is part 3 of the Chapter 20 lecture notes. On this slide, it's just an image that's trying to help you understand evolution doesn't necessarily lead to perfection. Organisms have been evolving for billions of years, and I don't think there's a perfect organism yet, as far as one that's going to live forever and perfect as in, you know, uh, has no flaws at all. Um, if you look at the evolution that led to the um, octopus eye and the human eye, okay, let's see. Number 4 represents a blind spot in our eye, and the octopus eye doesn't have that. But our eye works for what we need, even though the octopus eye is better. I mean, it looks very, very similar, but the octopus eye does not have that blind spot. So this is just one example, but it's important to understand that mutations, DNA mutations, are random. They just happen as DNA replicates. Every time a cell divides, DNA has to replicate. All the DNA inside of that cell has to replicate during the S phase of interphase. And during the replication process, mutations occur. They are random, and sometimes they are fixed by the process that we learned about in, I think it was chapter 15. Um, but DNA polymerase can sometimes fix the mistake. It can detect the mistake and fix it. Sometimes the mutation is a silent mutation and it doesn't result in a change in the protein. But the point is that mutations happen, and they happen pretty often. Many mutations are required to actually lead to a new trait. And that trait, even though we use that word mutation and it sounds bad, the trait may actually be beneficial, it may be harmful, or it may have no effect. It may be neutral. So it's important to understand what evolution is. Evolution is a change in organisms over time. That's the definition. Individuals do not evolve. We say that sometimes when we're talking, just in not about biology, but just in normal conversation. We say, I have evolved, meaning that I've changed over time. But the true definition of evolution is a change in species characteristics over time, not where an individual has changed over time. Um, if you have the giraffe, for example, this is a really common example of evolution, is that the older fossils of giraffes had shorter necks, and so evolution has led to this change over time in this really super long neck in a giraffe, so it can reach leaves higher in the tree branches. But it's not like one giraffe stretched its neck somehow and evolved a long neck and then had offspring, and those offspring had long necks. It wasn't like that. What happened is there were a group of offspring at one point that one of them had a longer neck due to a mutation in the DNA. And that one survived because it was able to eat the leaves higher up in the tree, and it passed on its trait. And over time, that happens over and over and over again. It's called natural selection. And after a while, you look around, and most of the giraffes have long necks. And over time, the short neck trait is actually breeded out. You know, there may be some giraffes born with a short neck if they have a mutation, but all giraffes that we see at this point in time have a long neck. So evolution is the change in the characteristics of a group of organisms over time, not individuals. Okay. All right. So Shared characteristics are used to determine relatedness when you're constructing a cladogram. Shared ancestral characters are found in the common ancestor of the group. So in this group, the vertebral column is a shared ancestral character. All the members of the group have it, well, except for the lancelet, the little lancelet, but a shared derived character, which is also known as synapomorphy, is derived within the larger clade. So it distinguishes those that share it from those that do not. So for example, a shared derived character would be the amniotic egg, and that is shared with the lizard and the rabbit. So all the other organisms on this list don't lay amniotic eggs. So a shared ancestral character is shared by all the organisms in the clade, and then the derived character is only shared by a few members of the clade, and it's used to identify the branch points within the clade. All right, now the rule of maximum parsimony. There is a study guide question on this, I remember. So parsimony just means that you choose the simplest way to make your cladogram, or maximum parsimony means we're choosing the simplest explanation. So this cladogram is less parsimonious because you've got B in two places. Now putting B down here is more parsimonious. It's simpler to do it that way because when you do it that way, you know that B is an ancestor to C and D. So it's simpler to draw it the way it is on the right. Let's see. I'm just skipping that. I'm sorry. Okay, so I'll talk about it a little bit, but the concept of phylogenetic trees started with Charles Darwin, and Charles Darwin, of course, is the scientist that discovered natural selection by studying. Particularly, he's famous for studying the animals that live and the plants that live on the Galapagos Islands. And he studied fossils and living specimens, and was able to determine that different species evolved by natural selection, adapting to, sorry, adapting to different environmental constraints. So we're only doing this one chapter on evolution. So I just want to say to you, I know students sometimes ask, and we're not in class, and I don't know what questions you may or may not have, but I personally cannot explain to you how evolution fits in with what we learn in Genesis of the Bible. Um, it, it's just, I mean, I have no idea how how to even start. I mean, you can Google and study that on your own. What I can tell you is the theory of evolution. Some of the evidence that's used, well, there's lots of evidence that the theory of evolution is accurate, for one thing. I mean, there's tons of evidence. Um, but we're still, the experiments that we've done we're still having to use them to determine what happened in the past, and we weren't here in the past. So what we have to do is use what we can see in the present to figure out what happened in the past, and what we can see happening in the present time, in the time since humans have existed and started making records, is that we have seen speciation within our lifetimes. We see new species form. We see mutations in organisms. We have seen, I can tell you one example is the resistance, bacterial resistance to penicillin. When I was a little girl, if you were sick, you went to the doctor and you got penicillin, and that was it. There wasn't a question. You take a child to the doctor now, and penicillin won't work because bacteria have become resistant over time. That is an example of natural selection, and that's what drives evolution. That's what causes evolution over time. So, um, how to get that to work in with your religious views, I don't know. I don't know how to do it, with my own religious views. I just know that this is what we have. This is the best theory. This is the best that we have right now as far as an explanation for how we got here. So, moving on to species concepts, there's the biological species concept, and then there's the phylogenetic species concept. Biological species concept defines species as groups of interbreeding populations that are reproductively isolated. That means they can breed with each other, they can mate with each other, and they produce fertile offspring. But phylogenetic species concept says a species is a population characterized by one or more shared derived characters. It has nothing to do with interbreeding. And the reason the biological species concept, there's a problem with that, is because this restricts the definition to those organisms that undergo sexual reproduction. But what about bacteria that don't? So, I mean, they just divide in half. They just basically clone themselves. So, the phylogenetic species concept works for all organisms, and it depends on what your what area you're studying, what area of research that you're doing, um, as to which concept that you're using. Okay, so the limitations to the classic model of inheritance, the Mendelian inheritance, is Mendelian inheritance assumes that genes are transferred from parent to offspring, which is vertical gene transfer. When genes are transferred from parent to offspring, and mutations are the sources of new genetic information. So a new gene is a result of a mutation. But in prokaryotes and even some eukaryotes, some single-celled eukaryotes, this is not the complete story. Prokaryotes, like bacteria, can actually transfer genetic material between unrelated species. A dead bacterium and a live bacterium, the a gene from, this is a little plasmid, which is a small circular ring of DNA, can be transferred from that dead bacterium into that live bacterium. Frederick Griffith showed us that, if you remember. So that's horizontal gene transfer. It's when, and this bacterium could be the one that causes strep throat, streptococcus, and this one could be E. coli. They can be completely unrelated and the genes be transferred from one to another. Now, there are three primary modes of horizontal gene transfer in prokaryotes, in bacteria and archaea. There's transformation, and transformation is what I just described to you. It's when the gene comes from an outside source, maybe a dead bacterium that's living in the same environment, very close to the living bacterium, and its gene is taken up by the living bacterium and it becomes a part of the genome, a part of the chromosome of the live bacterium. That's transformation. Transduction is when a virus transfers the gene to a bacterium. And then conjugation is actually when two bacteria, one actually inserts its plasmid, that small ring of DNA called a plasmid. It actually injects its plasmid into another one, and that's conjugation. So these are three ways that horizontal gene transfer can occur.